Thank you very much for spending all that time with me, and I'm learning a lot, and I hope this starting project with Blanca is going to be the beginning of many more. And thanks also in particular for the wonderful trip I had yesterday to the National Park, Cotonijana, seeing my first imperial eagle that I had sort of on my list since I was here more than 30 years ago in a neighborhood, but of course couldn't get near them at the time. So that's wonderful. So that's something I'll never forget. So I'm going to talk to you about telomeres and life histories. But I first want to say something brief about the background from why I'm interested in telomeres. And so my real interest is this. It's on the one hand, it's the link on the one hand between life history decisions and the state of individuals. So how does state affect the reproductive decisions of individuals? And at the same time, how do those reproductive decisions affect their state? Because there's a mutual dynamic interaction there, of course. And then this will be driven to a large extent by how state will affect fitness. The decisions how to reproduce, given their effects on your state, and how those state differences affect your fitness. That will be important in the evolution of the life histories. So the big question was, and still is to some extent, to a large extent, what is state? Because state is an important concept. Phenotypic quality will be another way to put it, if you like. So it's an important concept in our thinking, but making that operational and trying to measure then the phenotypic quality of the state of an individual is usually very difficult. So this is why I turned to measuring telomeres. So telomeres, here's a picture of human chromosomes, and you see some light bits at the end of the chromosomes, and those are the telomeres, and they consist, so here's the same thing as in a cartoon form, and they consist of this sequence TTA-GGG, which repeats itself thousands of times, or in the case of some bird species, it can be, say, up to 250,000 base pairs long, or even more. Yeah? They are complexes together with a number of proteins, and they end in a telomeric loop, in a single strand telomeric loop shown on a picture here. Yeah? This single strand loop is important in a practical sense because we use that to measure telomeres by binding probes only to this telomeric loop. So because every chromosome has a telomere and every cell has lots of chromosomes, it's not so that you can characterize the telomere length of an individual, say, with one number, because it's actually a whole distribution. You can see that here is a picture of my technician, Alice Mulder, who does the magic with the telomeres that I only know about in theory. And what you see here is the telomeres on a gel, so each lane here contains one sample. And what you can see, this is not just one band, but it's a whole smear. And this is not measurement error or anything imprecise in the technique. This is so because the telomere length of an individual is not, as I said, is not one point, but the whole distribution. And that is shown here in more detail with a new technique to measure telomeres. So this is a gel made for us of a jackdaw sample by Tsung-Po Lai in the States. And what you see here, so actually all these lanes here, they're all lanes of the same DNA of the same blood sample. And each band you see here, and all these here as well, they are all single telomeres cut from a single chromosome. Yeah, so I'm showing you just to emphasize that there is not one telomere length, but when you measure it, you get the whole distribution, or when you use qPCR, you get only one data point. But this is actually what is underlying that. Yeah. Okay, so that's the first thing I wanted to tell you about telomeres. So what they are. An important characteristic of telomeres in most species in which it has been studied is that they decline with age. So this is quite a large sample for humans. You see age on the x-axis here is a huge range, and you see this decline. You see an enormous amount of scatter also. This is caused by variation between individuals, which is typically very large. So this is usually it's about the 3 kb range. So 3,000 base pair between the individuals with the shortest and the longest telomeres. In bird populations, we also see typically this very large variation between individuals. I'll come back later to the source of that variation, or at least part of the source of that variation. So telomeres length typically declines with age. Now, this does not happen in all tissues at the same rate. It has to do with the rate of cell division in those tissues. So usually, this is in humans and in other mammals, of course, it's usually just the white blood cells that are used for telomere measurements because it's relatively easy to sample. Of course, in birds, we can use the red blood cells because they're nucleated, which is an important difference between birds and mammals. So as I said, the rate of telomere shortening with age is not the same in all tissues, and it's shown here in another recent paper of us, where you see on the x-axis, you see the telomere length measured in muscle cells, and I should mention that all the human data I'll be showing were not measured in our lab, but in a lab of a collaborator in the States, in Newark, Avi Aviv. So anyway, so here you see the muscle telomere length, and here is the telomere length in blood of those same individuals, so different tissue. And what you see here on the top here is the measurements in fetuses. So they are from abortions, spontaneous or otherwise. And this line is the line of equal values. Yeah? So when the points are on the line, it means that telomere length is the same in blood as it is in muscle. And you see already in fetuses of a couple of weeks old, you can see that telomere length in blood is a little bit shorter than in muscles. Yeah? If you then look at children, you see on the one hand that the shift is to the left or down on this axis. So telomere length both in muscle and blood becomes shorter over this period of development. But the gap between this line and the data point increases. So what that means is that telomere length in blood shortens at a higher rate than telomere length in muscles. And this is useful to remember because I'm going to use this, we're going to use this insight at a later point in the talk. So this is then in adults, where you see that the distribution here is shifted to the left and shifted down, but again the gap between this line of equal values has broadened, just showing that also that throughout life actually telomere length shortens at a higher rate in blood than in muscle. So almost all the data I'll be showing are actually on telomere measurements in blood, but it's useful to keep this in mind. So the question I asked when thinking whether I can use telomere length as an estimate of the state of an individual really boils down to, so to what extent are variation in life histories then synchronous with variation in telomere dynamics? And I'm thinking of individual variation in life histories here more than interspecific variation. So another way of asking this is to what extent does telomere length predict fitness of individuals, and where we can look at that separately for reproduction and survival. Yeah? So that's what I'll be doing in the next part of the talk, starting with survival or mortality, you can say in this case. And this is actually what started me being interested in telomeres. This publication by Rick Cawthon, where he showed that if you plot the years after blood draw on the x-axis and the proportion of surviving on the y-axis, then those individuals with longer telomeres, you see they survive slightly better than individuals with shorter telomeres. Yeah? If you correct this for age, this stands. Since then, many studies have come out, some supporting this and others not, but on the whole it's clear that there is this association, and I think we are in general convinced of this, that this is the case. We actually published a meta-analysis a couple of years ago showing this. We now have what is probably one of the best data sets that we're still working on to show this. So this says age on the x-axis and this is survival on the y-axis, and these are data from 3 long-term studies in the States that are really well known. These people are really studied in detail. So when someone dies, a full committee will decide what was the cause of death. That's how detailed the study is. What you see here is that, so this is the survival of these cohorts, and the top line here is these individuals, so the 25% of individuals with the longest telomeres, and this is the 25% of people with the shortest telomeres, and you can see that their lifespan is clearly affected. What we see also in this graph, and this is actually the first time that data are sufficient quality to say something about that, is that the effect seems to level off. So whether you're in the top 25% or in the next 25% doesn't seem to be much difference. But when your telomeres become shorter than the median, then it quickly starts to deteriorate. Yeah? So it seems to be a nonlinear association. So this is in humans still. Also in birds, there's one thing that there was a paper quite a while ago by Mark Hausmann and others showing that in bird species that had a short maximum lifespan, telomere length declined faster with age than in species with a long lifespan. And actually last Monday we published a follow-up of that study led by Mark, and this shows this for a much larger data set and also with a with an analysis corrected for phylogenetic relationships. What you see here is on the x-axis is the telomere length rate of change. So when it's high, it means telomeres decline with age fast. And when it's low, it means they are relatively stable. And on the y-axis is the maximum lifespan. And what you see across these set of species, which were all measured in about the same way, either in Mark's lab or in ours. I think it's just those two labs. And you see there's this decline. So this further indicates at another level an association between telomere dynamics, telomere length, and lifespan and mortality. Yeah? So this is fairly well established. So I'm going to leave the survival part at this and try now to say something about reproduction. Now, this is a bit more interesting in a sense, and the reason is that not only can one expect or look for effects of telomere length on reproductions, or maybe I should say associations between the two, because I don't want to suggest any causality. But at the same time, it's possible that your investment in reproduction has an effect on telomere length. This is also what I sort of described in my first slide, looking at this interaction between state and life history decisions. We collected some data from the literature on this to see what people had found in general. And of course, one way is to look at this is cross-sectionally. So that means going into a population and taking blood samples and looking how well reproduction was. And I should say that this slide is now 1 or 2 years old or so and should definitely be updated, but the general picture doesn't change much, so that's why I still dare show it. So what you see is that there tends to be in most species a positive association, but it's significant in very few of them, which may in part have to do with this large individual variation in telomere lengths that I showed you in the beginning. Here's a very interesting exception. It's the common tern, where individuals with long telomeres actually fare poor at reproduction. They fare poor in lots of aspects. So they come back later to the breeding grounds, for example. So it already starts with that. Yeah? So this is, if you like, this is this part. Now that was the cross-sectional analysis of something about. But we can also look longitudinally and say, so what happens? So individuals that were successful at reproduction, what happens to their telomeres when we come back, say, a year later and sample again? And then we see interesting variation again. So here, these three species, there's a trend towards those individuals that were more successful losing fewer base pairs. So they do better. And for the gray tit, and I'm going to come back to that because it's one of our own studies, there it's actually a statistically significant effect. But again, in the common tern, it is those individuals that were more successful at reproduction, they actually lose more base pairs to the next year. So I just want to come back to these two studies. So this is how we show that, and this data set is quite small, but also over a much larger data set we actually find the same effect. So you see here telomere length in year 1, and here telomere length in year 2. And this is the line of equal value, so if there's no change, they're on that line. Yeah? And the closed dots here, they are the birds that lost their chicks really very early in the season, so they didn't work very hard for their offspring in that year. While those individuals that raised their chicks for a much longer period, usually to fledging, there's the open dots, and you can see that they lost more base pairs, and it's a difference of 257 base pairs, and that's quite a lot. That's about, I think it's about 3 times what they lose on average, but I'm not quite sure. So this is a clear difference where the individuals that do best, they actually lose most base pairs. And the great hits, we find the opposite. And this graph is a little bit more complex than necessary, so you can ignore this part, and this is the estimate that matters now here. And it just shows you that those individuals that were more successful, that they lose fewer recruits. Take my word for it, for now. Yeah? They lost fewer base pairs. So and this, I think this hints at something very interesting, something sort of a more general question to which we don't have an answer, which is not so much related to telomeres, and that is that if you look at different bird populations or different species of birds, and you try to find out which individuals are more successful, then we don't actually know how that varies between species. Our interpretation of this is that in the terns, it is those individuals that can work hardest are the individuals that are most successful, and they pay a price in losing these base pairs. And of course, they all forage in the same area. They don't have a territory. They all fly out to sea and forage there. So it's really who is best able to get fish out of the sea, which can be a matter of knowing where to go, but also, of course, how fast you can fly and how many hours you can keep it up. So those individuals are most successful in the terns. But in the great hits, it's maybe those individuals that don't have to work so hard that are most successful, because they have a very good territory. Because they get their food from the territory, it may be that determines which individuals are most successful. So I just want to point out that, and I haven't thought about this much longer, or certainly not collected any data on it, but we do not really know very well how it varies between species, what makes individuals successful. But I think that's an interesting thing worth thinking about more. Okay, so of course these are correlational data, in the sense that the reproductive success was not manipulated in the sense that we made some individuals work harder and gave other individuals an easy life. Because that's of course where we would see that's what was shown here, what comes out when you do that. So the Adelie penguins was a sort of a handicap experiment, and so was the great tit, and you see very little effect there. And this is actually, so this is our own project. This is about this is many dozens of birds that had to carry a weight of 0.8 gram for a whole year. And you see there's no effect at all. So it's quite convincing to us, at least, that there's no effect. Here are 2 brood size manipulations, and in particular the zebrafinch is interesting that it's sort of a short-term effect, and it then persists, but if you keep letting them reproduce more, it doesn't change much. So the link between reproduction, to me, is still sort of half clear only. We did an experiment of, we had an experiment to look at this ourselves in jackdaws. So the jackdaws, they breed in nest boxes that are in colonies, and it's a bit too dark to see, but you see several nest boxes on these trees, and so they're very accessible, and it's a nice thing with these colonies. You can actually, it's a very efficient way to collect data. And what we did is we did a lifelong manipulation of reproductive effort. So what we did in every year is that we manipulated the number of chicks in the nest, and we had no control broods, just enlarged and reduced for sample size reasons or for reasons of statistical power. So this is what you see after the manipulation. And it's a lifelong manipulation in the sense that what we did there is that when individuals were assigned to rearing either reduced broods or enlarged broods, they would be doing that the rest of their life. And so we identify them before their chicks hatch, and then we know already what manipulation they'll be receiving. And what we were able to show with this, and maybe this graph is the most easiest one to interpret. So here is the age of the birds in the x-axis, and here is their mortality rate, so their instantaneous mortality rate over those years. And what you see is that those birds that were forced to rear enlarged broods every year of their life, the rate of actuarial senescence, so that is the rate at which mortality rate increases with age, is steeper in the birds rearing enlarged broods. And this is sort of this is the plot of the posterior distributions of the parameters, yeah, of one of them. So this shows that we experimentally accelerated the rate of aging on a demographic level, and of course now we wanted to know whether that also accelerated the rate of aging in the state variable that we're looking at, namely telomere length. And this, much of this of the telomere analysis, so much of the experimental work was done by Jelle Bonenkamp, who was a postdoc and a PhD student with me, and much of the telomere work was done by Christina Bauch, shown here, sampling a jackdaw in the back of our field car. So she analyzed how this manipulation that we now know affects mortality, how this affects telomere dynamics. And I just want to briefly say again that the value of that we know that it affects mortality, because costs of reproduction are actually more often not observed than observed when it comes to survival cost in particular. And in a meta-analysis, pooled overall studies is not a convincing effect at all. So that means that whenever you look at the physiological variable or telomere length, in the context of a brood size manipulation, thinking you're investigating the mechanisms behind the cost of reproduction, that sort of assumes that there is a cost of reproduction in your set. And it turns out quite often that's not the case. So it means that you're studying rather adaptation to the physiological adaptation to the changed circumstances rather than the mechanism mediating the cost of reproduction. So the fact that we have this cost of reproduction here, we consider quite important when looking at physiological variables. Yeah. So she looked at the telomere dynamics of these of a subset of these individuals, and we sort of have decided that actually we're going to need to expand the data set for different reasons, but it's not going to change the picture very much, which you see here. So this is the number of years in manipulation, and this is the telomere attrition. So this is how much telomeres they lost since we first started manipulating that individual. And what you see is that enlarged broods and reduced broods, they're very similar. Of course, the sample size here at this end is quite low, and there's no evidence at all that we affected the telomere dynamics by having individuals reared up to, say, 8 enlarged broods over as many years. Yeah? So it's quite substantial. What you did find is that, as we found before, that there was quite a clear association between telomere length and survival, but this was affected by the manipulation. So you see this here. You see here this telomere length it's corrected for age, so that's why there are residuals. And you see that in those birds rearing reduced broods, the telomere length of those birds that returned to breed in the next year was only slightly longer than the telomere length of the birds that didn't return to next year. You can see the standard error is quite large here because many returned, so the sample size is not so high. Those birds rearing enlarged broods, the difference is much larger. So those that survive have much longer telomeres than those individuals that didn't. So a schematic way to plot it is shown here, that if you plot telomere length in the x-axis and rate of survival on the y-axis, then in those birds that have an easy life with reduced broods, you can see that the selection on telomere length is relatively mild, while those birds that rear enlarged broods, it is much steeper. So telomere length is clearly important in this experiment, in that it mediates who dies in the enlarged group, but the manipulation doesn't affect telomere length. Okay. So having established that there are these clear associations between telomere length and different fitness components in different ways, the question arises, so what causes then variation in telomere length? What makes some individuals have much longer telomeres than another individual? And one thing that's useful to know here, and this is, appears true in humans as well as in birds as far as we can tell, is that telomere length seems to be determined very much early in life, in the sense that telomere length declines with age, but individuals that have short telomeres for their age when they're young, they will also have telomeres short for their age when they're old. And it's shown in a sense here. So this is the telomere length of nestling jackdaws on day 5, and this is the telomere length before they fledge, just before they fledge when they're 30 days old, and you see it's a very tight correlation. Yeah? It also illustrates the quality of the telomere measurements of my technician. So I take no honor for this at all. It's her work. And if you plot then the telomere length of the birds when they fledge against their telomere length when they return 2 or 3 years later, you see there's quite a tight correlation, and Christina Bauch has now expanded this data set, and so the R square there is about 0.75. Yeah, so the telomere length with which individuals fledge seems to be, to a large extent, not fully, but to a large extent, seems to determine the telomere length they have throughout their adult life, corrected for age. Yeah? That's shown here as well by slightly. So this is on the x-axis here is the telomere length when individuals were first caught as recruit. So we knew their age exactly, and we had them as nestling. And on the y-axis is the telomere length the last time we measured them. Yeah? And there can be 1 to 7 years between those first and last measurements. And what you see here, and they're given in different colors, and what you see that this correlation is always very tight. It just confirms the point I just made, that individuals that have a long telomere length for their age, they will have that for life. And it's also shown by the fact that, well, you see this line is sort of really below, so it's decreased most over the years, but the correlation remains as tight as it was. Yeah. So for the jackdaws, it appears that the telomere dynamics is really not very variable. There is actually, I'm not going to show that, but there is a little bit of variation between males, and at least in the rate at which they lose their telomeres. But you can see it's very small scale, and you need very precise measurements to be able to say anything about it. Okay. So we now know that telomere length seems to, if you correct this for age, seems to be sort of fixed for life, although there are some, as I'll make show later, there's some modifications in early development. So what causes then this variation? And on the face of it, the heritability of this seems to be very important. And so there's a number of studies showing this, and in humans it's about 0.66, the heritability across studies, but actually it ranges widely across the different estimates. We looked at this in the jackdaws, and we started with simple parent-offspring regression. So this is offspring against father telomere length, and offspring against mother telomere length. And you can see that there's reasonably strong correlations. But of course, here is the problem that we have a shared environment between parents and offspring. And so the extent to which that explains the association, we cannot tell from these data. So for this reason, we did some slightly more sophisticated work using the pedigree of our telomere measurements, and these are almost 700 measurements of individuals, all at the same age, at day 5. So we don't have to consider age of the nestlings in these analyses. And when we estimate heritability using animal models in these data, we find a number of components. So the rearing nest identity. So the y-axis here is the variance explained by the different factors. The rearing nest identity doesn't seem to be very important. There's a small year effect. There's a small mother effect. But in particular, at about the same level as in humans, about 2/3 of the narrow sense heritability seems to be the narrow sense heritability seems to be about 2/3. Yeah? So that's very similar to humans. Now, telomere length inheritance is an interesting thing compared to many traits, slightly more interesting than many traits. And the reason is that there is potentially a sort of a dual inheritance mechanism. So there are two inheritance mechanisms. And that is firstly because inheritance of the genes that regulate telomere length. Yeah? So during early development, telomere length is regulated through telomerase, an enzyme that can elongate telomeres, and it's usually active for quite a short period in life. In humans, it's inactive very early, a few months, I think. And so that is one mechanism. But on top of that, the zygote inherits directly the telomeres from the gametes of its parents. Yeah? So what is quite unusual is that the phenotype is already expressed in the zygote on day 0. Yeah? And that is not true for many phenotypes, none that I can think of, actually. And the question, of course, is, does it matter? Is there then a carryover effect of that telomere length in the gametes, so that's the parental phenotype, if you like, through adulthood, or is it regulated away? So schematically, I've drawn it like this. So this is a zygote that started with short telomeres in the gametes, and this is a zygote that started with long telomeres in the gametes. And over development, telomeres will shorten. I showed you this already in the human data, from the fetus to children. There was already quite a large effect. But what you see here is that in this schematics, there is some compensation. Telomeres that are long tend to shorten at a slightly higher rate. But even at birth, still those that started out with longer telomeres because of the gametes, they still have slightly longer telomeres at birth. Alternatively, it's only a transient effect. So that would be the case when the telomere length regulating genes dominate the outcome fully and compensate for any differences that there are between gamete telomere length. And in that case, the thing would be the same, but except that here the compensation would be larger, and in the soma at birth, telomere length would no longer be different. So those are sort of the two possible schedules. And of course, if this is the thing that's happening, that would be sort of a form of Lamarckian inheritance, which would be fun if we could show that. So that's the idea that an organism can pass on characteristics that it has acquired during its lifetime to its offspring. Yeah? And there is one reason to think that this may be going on, and that is that in humans and in, I think it was chimpanzees, yeah, it's shown that when the father is older, the telomere length of his children is longer. So it's an increasing effect. And this is true both in chimpanzees and in humans. And for the chimpanzees, we know not much else, but for the humans, we actually know that telomeres in sperm is longer in older males as compared to younger males. Yeah? What we do not know yet is to what extent telomere length in sperm directly explains variation in telomere length in the offspring at birth. There was a paper out 2 weeks ago or so in salmons by Neil Metcalf and his group, and actually it wasn't so direct because you need a lot of sperm to be able to measure telomeres in it, so they had to pool things and so they did not find a direct association, but we know that at least in the zygote there must be a direct effect. So this is what is hypothesized explains the paternal age at conception effect in humans. Now, there are some problems with these data, however, and an important one to us is that all the data are cross-sectional, and so you can imagine that these are all different fathers that have children at different ages, and their telomere length among their children is longer with the increasing age. But it could be that if you follow all those fathers over time, that in fact this is what happens. It could be that within fathers, actually the effect goes the other way. So to what extent we really, the extent to which we know that this correlation between paternal age and telomere length in the offspring really is due to age, or to fathers with different telomeres having offspring at different ages, we don't know. We know that in women, their fertile part of their life is longer when their telomeres are longer, for example. So it's not unthinkable that there is such an effect. So anyway, the jackdaws, as you see, we have data for over quite a number of years. So there we could check whether there is a longitudinal effect of paternal age on the telomere length of the offspring. And this is what we find. So this plot shows a longitudinal change, because what is shown on the x-axis here is what we call delta age. So what it means is that for every father we say, so what is the mean age at which you sired offspring in our data set? And we express the delta age is then just the years after the mean and before the mean. So if we have offspring of a father when he was 3, 4, and 5 years old, then the first year would be -1 on this axis, and the last year would be +1, and the middle year would be 0. So this really shows what happens within individuals. And what you see is not a negligible decline of 75 base pairs per year in the offspring of the father. So we do find a paternal age effect, but it's opposite in sign to what is found in mammals, at least the 2 primates in which it has been investigated. Yeah? Again, of course, there is the question to what extent this could be due to some kind of environmental effect, in the sense that older fathers maybe take care of their offspring less well, or maybe they take better care of their offspring, so they grow faster and they lose more base pairs even over those few days, or those things are difficult to exclude beforehand. So what we did is, so this is a question we needed to resolve. And the way we address this is that we exchanged clutches between males with different age very soon after the clutch was complete, so that at least the period sort of between conception and sampling, which is when the nestlings are 5 days old, that we know that the father can have had no direct effect over that period. And what we see then is that, so these are the plots here, they're all on the positive side because we only did this the last 3 years. You cannot see this actually except that they fit in very well, but then if we estimate the slope separately for this, for those cross-fostered males or offspring and non-cross-fostered, we see that the slope among birds raised by foster parents is virtually identical to the slope among nestlings reared by the biological parents. Yeah? So there's no effect at all of cross-fostering, and thereby we assume that the effect was already present in the egg, if you like, and not through to a difference in parental care. So we think that this is evidence in support of the idea that there's a sort of a direct effect of the father's phenotype on the offspring phenotype, independent of parental care, which is already present at birth. Another way to plot this is like this, and that is that, so in the x-axis here is the delta telomere length of the father. So delta here is, so 0 here is the mean telomere length of the father over all the measurements we have of those males over its life. And so this will be the early years when he had relatively long telomeres, and this will be the late years when he had relatively short telomeres. And so here on the y-axis is the telomere length of the offspring, and again 0 here is the average telomere length of all the offspring that father produced over his life, at least those that we sampled. And so the deviations show here, so the deviations from the averages show a strong positive correlation. So in the years that the father had long telomeres, his offspring had long telomeres, and in the years that the father had short telomeres, also his offspring had short telomeres. So we tend to think that there's really a Lamarckian inheritance here of telomere length. That's what it looks like. Okay. So now I've shown you that telomere length are associated with all kinds of fitness components. At the same time, they seem to be to a large extent heritable, and in addition there is this epigenetic effect. And from an evolutionary perspective, it's actually not so easy to understand why a trait that is so strongly heritable can at the same time be predictive of fitness, because you would expect that, this is an old Fisher's prediction, you would expect that the genetic variation would erode for traits on which there is a lot of selection. And of course this hinges, to ask this question very much hinges on the extent to which we think that telomere length is actually causally involved in all those patterns that we see. And so the next thing I want to address, and the last thing I think, is I want to address is to what extent the patterns we see between telomeres and telomere dynamics, with telomere length in particular, may be causally related to the different fitness traits at which we've been looking. And so, because there's sort of two options, if you like, to put it simple, is that on the one hand, telomeres can be part of the causal chain when life stress sort of has a direct effect on telomere length, which has a direct effect on senescence or mortality or whatever fitness component you're looking at in particular. Alternatively, various forms of life stress can have an effect of telomeres and an independent effect on senescence, which would of course mean that there was sort of another state variable sort of here intermediate. Yeah. So those are sort of the extreme things. And of course, in this case, it could be that it doesn't matter so much that telomere length is strongly heritable, because there would be no direct selection on those genes coding for variation in telomere length. But in this case, that will be different. Okay. So there's some evidence that at least in some cases, telomere length may be causally involved in causing mortality. But it may be only in extreme cases. So what we see here is human data, and you see here telomere length, and you see here age, and the open dots are sort of control population. Actually, they're control relatives in this particular case, because the closed dots are carriers of a gene causing a disease called dyskeratosis congenita. And what you can see, it's a telomere disease. In fact, it affects the telomerase gene. You can see that they have very short telomeres. So this is something we can use to say, okay, so what really happens when telomeres are very short? Because when there would be no effect at all on these people, then we would know that the real length is not so important. But that turns out not to be the case. This is a survival plot of people, of carriers, and what you can see that at age 40, only half are still alive. Yeah? So, and this is really a disease sort of on telomeres. It's not sort of a side effect of something else. It's really direct related to the telomere length. So what this means is that there can at least be, in extreme cases, because these telomeres are very short, there can be a direct effect of telomere length on mortality. That's what it looks like. There's a mouse knockout line where telomerase is knocked out, and actually that supports this finding in the same way. I'm not going to show those data. Another way to look at whether telomere length is causally involved is the following. We can look at the association, say, between mortality and telomere length in different ways. It could be that when telomere length is only informative because it's an index, say, of damage, then we would say that the telomere shortening should be most important. And if that were so telomere attrition, if it would so, then if we would have two individuals sampled at two different ages, and their telomere length declines, and in this individual it starts out with relatively short telomeres and it doesn't decline so much, and in this individual starts out with long telomeres and it loses a lot of base pairs, so they end up with the same telomere length. If the telomere attrition was important, we would expect this individual to have a much higher chance of dying than the other individual. And that is something we can test. Yeah, we can do that. So the absolute length is the same, but still there's a difference in survival probability. That would mean that attrition is important, and that would support the idea that there is no causal effect of telomere length. Because when there is a causal effect of telomere length, then if we were two individuals that lose, say, an equal amount of base pairs, but the one that has the lowest number of base pairs left is the most likely to die, then that would fit the idea that there's a causal relationship. It doesn't show it, but it would be in agreement with that. So we looked at this in jackdaw nestlings, and we compare between birds that recruited, so those are the closed dots, and those that didn't recruit. Those are the open dots. And first we had the telomere length just after hatch on day 5, and you show there's no evidence at all for a difference. Yeah? If we then sample them again at day 30, there is a hint that there is a difference. This is actually, no, I don't have to explain that actually. So there is a hint there's a difference, but it's very far from being statistically significant. Now if we then compare how much telomeres they lost between day 5 and day 30, so that's delta telomere length, we see that those individuals that died, that did not return at least to be recruited as breeding birds to our population, that they lost 280 base pairs, while those that made it lost only 200 base pairs. So this fits the idea of telomere length or telomere dynamics being an index of what has happened to the individual, rather than telomere length having a direct effect on survival. We recently showed this in a very different way also in humans, not with respect to mortality, but to arteriosclerotic disease, so arteriosclerosis. So I'm going to take you this step by step. So what is well known, if you look at leukocyte telomere length, so that's in the white blood cells, and you compare individuals age-corrected without disease and with the disease, they have shorter telomeres with the disease. This is actually this is one of the first things known about the association between telomere length and health in humans. So this is in blood. This is in blood. If we also look at this in muscle, we see no difference at all. Yeah. Now, as I said at the beginning of the talk, the rate of change of telomere length in muscle is slower than it is in blood. And because of that, the telomere length in muscle better reflects telomere length of individuals in early life than the telomere length in blood. Yeah? So what we tried here is to say, well, perhaps if we express the telomere length in blood relative to the telomere length in muscle, we have an index or an indicator of how much base pairs were lost in blood, yeah, over that time. And we did that in 2 different ways. So this is the absolute difference, and this is the ratio. And what we see is that it's not surprising given these data. What we see is that the difference between the two is larger in individuals with the disease. Yeah? Now, this in itself, of course, is logical, but what is more important is that we did a model selection procedure, and what we can show is that if you express telomere length in blood relative to telomere length in muscle, it gives a significantly better prediction of who has arteriosclerosis and who doesn't. So the fact that it's not the absolute length, but the relative length, supports the idea that it's the telomere attrition that's important, and not the absolute telomere length. We could carry this one step further, so this was recently published if you want to read up on that. If we plot this difference between, so it's this difference. So if we plot the difference in telomere length between muscle and blood against age, not surprisingly, it increases, because it continues to be the case that muscles lose fewer base pairs per year than blood. Yeah? What is important is that these lines run completely in parallel. So what that means is that the difference, and you can shoot the slopes here, so this 0.015 and also 0.015, really they're really about as similar as they can be. Yeah? So what this means is that the difference in telomere length between individuals with and without arteriosclerosis is already present at the beginning of adulthood. Yeah? So as in the jackdaw nestlings, it seems to be the telomeres that you lost during your early development that determines whether you have this arteriosclerosis or not. And actually, we all get it if we live long enough. So really, it's more about the age at which you get it than whether you get it at all. Yeah, you just get it to a different degree. Okay, so this is this sort of fits what I showed at the jackdaws, that there also the telomere length seemed to be determined early in life. But although the differences sort of remain quite persistent, we could see that how much base pairs they lost during development was an important predictor. And you need, it's when you think about doing this yourself, it's worth being aware that you really need very precise estimates to be able to say anything about this. And then it seems to be really that what happens sort of before you're 20, when you're human, that has a strong effect on well, in this case, so cardiovascular disease-related pathologies. Yeah? Okay, so coming back to this question on the causality, these are two sort of extremes, and of course the mixtures are possible. My view, at least, and I'm well aware that not everybody agrees with this. This is sort of an open question in the field, but my view, at least, is that telomere lengths are causally involved in extreme cases, and so they can also be causally involved, say, when humans become very old. But in the general population, I think that telomere shortening is more an index than it has a direct effect. And the big question now is, of course, is what is it an index of? Because now we can use this, say, for example, in the jackdaw nestlings, and say, okay, so what determines then which individuals lose few base pairs and which individuals lose as many? And we can try and do that as a physiological level also. So that's not just an ecological one, but what is different in the physiology of those individuals. So that's one of the things that we are studying. Okay. And that's the last thing I wanted to tell you, I see. So thank you for your attention. And these are... Of course, I'm talking about a lot of people. I just realized I've forgotten all the people involved in the human work, which, anyway, would be a huge list. And I'm happy to take any questions. And at the bottom, you see that all the PDFs of the papers can be downloaded. Just don't tell anyone.